and we're grateful to have him here today to talk to us on what dairy farmers should know about genetic selection. So he'll be answering many of the questions we just had. Take it away.
Thank you very much, Mary. As I was listening to each of those questions, the answer is in this presentation. And if it isn't, you can ask it again. So some of these slides are similar to what Francisco had. I don't think there's any that disagree with anything that he had. So some of these I'm going to pass over relatively quickly because they're a little bit, maybe a little bit, some of them a little redundant. But this shows a little bit the same slide that he showed. In 1940s, we had about 25 million dairy cows in the United States, and they produced about 4,000 pounds of milk. And over time, we've increased milk production and decreased numbers. Today, we have about 9 million dairy cows in the United States, and they produce more than twice as much milk as the 25 million did back then. So the carbon footprint of dairy cows is about 43% less, while producing twice as much product. So again, not all of this is due to genetics, although we take our fair share of the credit. But better feed, better management, taking better care of the cows, more cow comfort, better feed quality, and all the kinds of things we'll hear today contribute to part of that. But as Francisco showed, over time, incredible increase. This is the genetic increase, very similar to the phenotypic. As we talked about, the protein yield is about 83% correlated with milk, so no surprise that genetic trend follows on about the same. Productive life, we didn't include it in the index until about 2000 or thereabouts, and so you can see that since it's been included in the index, we've made tremendous progress. Somatic cell, again, because we want to lower somatic cell. When the genetic trend goes down, that's a good thing. Again, we didn't include somatic cells into the index until about right here, and as soon as we did, dramatic change for the better. Daughter pregnancy rate, Francisco talked about genetic correlations between traits. There's a strong negative correlation between production and reproduction. As you increase production, reproduction generally suffers. And in the United States, because we're on such a path of increased production, the daughter pregnancy rate declined quite severely. And again, in 2003, we added daughter pregnancy rate to the index, and see what a dramatic change we've made in just a fairly short amount of time. So genetics does work. Selection indexes work. Here's the proof in the pudding. Here's when we added the traits. Amazing. In 1926, all we measured was fat and protein. I mean, milk and fat. We didn't even add protein until 1978. Daughter pregnancy rate. You can see when we added some of these other traits. The question is, what traits might we add in 2016, 2018, 2020? And there's a whole host of candidate traits that we'll be adding in the next few years. So most of the traits that are of economic importance in dairy are controlled by additive genetics, and so you must continue to add superior genes every generation to keep up your genetics moving forward. And a little bit like Francisco talked about, they're permanent, but you need to keep adding those good genes each generation. So why the service sires you choose for use in your herd, why is that more important now than ever before? I think the answer is because of genomics era. The rate of genetic gain is moving so quickly that if you're not moving your genetic standards forward by selecting better bulls for whatever economic traits are important to you, the efficiency of your operation is going to be left behind. So there are four pathways in which we make genetic progress. The dam of sons, the sire of sons, the dams of daughters, and the sire of daughters. There are few elite herds, of course, are going to have the cows that produce the sons. There are going to be a few sire of sons, and so these genetic pathways, by far, the most genetic progress is made for sires of sons. The second is dam of sons. Dam of daughters, unfortunately or fortunately, we breed almost every one of the cows in our herd that are available to breed, and so genetic selection on this pathway of dams of daughters is very low, and so the amount of genetic progress you're going to make through that is very low. So for most dairy farmers, this is the two pathways that they have available to make genetic selection. There's very little genetic selection here, so almost all of your genetic gain will come through the sires that you choose and their daughters that are from them. This is what we think of in terms of traditional pedigree. We've got the sire, the dam, and the four grandparents. And if we had two animals that had exactly the same parents, we would say that their parent average is exactly the same, because we assumed that they received a random set of genetics from their parents. So they would have the same genetic parent average. Under genomics, we know that assumption can be very, very wrong. This is how we think about a pedigree in terms of genomics. This happens to be for a single chromosome, but here are the four grandparents, and that chromosome split right there, and so half of this sire is represented by his mother's genetics and half by his sire. But in this chromosome, it's split right here, and we have most of this female is due to the genetics of her mother from this chromosome, and then it's split again. Notice here that this sire and this sire are completely gone. There is no genetics in this animal on this sire side from those two grandsires. Well, here, because of the way that's split, all four parents are represented. So now we could have a situation where two full brothers could be 300 net merit points apart, basically because the chromosomes split in different places. The other thing is that we can track an animal back to its grandparents and great-grandparents. We can also track the breed purity. How much of this animal is Jersey versus Holstein versus Milky Shorthorn versus whatever other breed. So genomics has changed the game, the way we think about pedigrees and the way we think about animals quite significantly. Genomics testing has been taken up extremely a high rate in the United States. In May of it started in January of 2009, basically. In May of 2015, the 1 millionth animal was recorded in the genotype database. And in August, the 1 millionth animal was genotyped. Notice that 184,000 bulls had been done, but 829,000 females. I can tell you that there's some other major dairy countries in the world where they do about 30,000 females a year. In the United States, we're now approaching 1 million females here So again, the way that you make rate of genetic change is, here's the formula. It's the selection intensity times the accuracy of selection times the standard deviation divided by the generation interval. And what has genomics done for us here? The selection intensity has gone from moderately low to extremely high. 184,000 bulls have been tested, and we only put into active service about 12 to 1,500 bulls a year. So 20 bulls they're highly selected to start with. Then you test those bulls, about 30,000 bulls a year, and then only 1 out of 20. So the selection intensity has gone extremely high. The accuracy of selection, back on the pedigree, we had an accuracy of about 35%. Now the accuracy of a genomic pedigree is about 75%. So the accuracy of selection has changed, and generation interval is almost staggering what has happened. Before genomics, the average age of the sire that was used to sire the next generation was 85 months old. Generally, we didn't use a bull very extensively until at least he was 5 years old, progeny tested with 100 to 200 progeny. And so, and the females are a little bit the same. But in 2013, the average age of both the sire and the dam have collapsed to 33 months. And we've been more increased genetic selection since that time, and so the average age of the parents are almost reaching biological limits about how young they can be. So the rate of genetic gain, because 3 of those 4 factors have significantly changed, the rate of genetic gain from 2000 to 2004 was about $20 of genetic change per year. From 2005 to 2009, it increased to 47, but from 2014 to 2010 to 14, the rate of genetic change is at $87.49. So again, we're on a fast track of genetic superiority. Again, from 2005 to 2008, compared to 2008 to 2013, the rate of genetic change for traits like milk, fat, protein, the indexes of net merit, food merit, and cheese merit doubled. But the important one is on the low heritable traits, like productive life, daughter pregnancy rate, and some of those, the increase was almost 3 times the rate it had been under before genomics. Here's some examples, and we get the genetic evaluations 3 times a year. So the genetic evaluation for April 2016 came out yesterday. So those of you that have got your slides, you'll notice that I updated from December, updated the slide as of yesterday for the new genetic evaluations. As Tom Lawler said, hot off the press, here's the values that virtually nobody had. But last year, the best young bull that got his genomic evaluation for the first time in April was 2,691 pounds of milk. Yesterday, a bull came out with 3,288 pounds of milk. So you now have, of course, these bulls are too young to be producing semen. These are. But 597 pounds of milk increased genetic value in 1 year. 21 pounds increase in fat, only 2 pounds increase in protein, but 172 pounds higher in net merit and 102 TPI points higher for the bull born this year versus bulls born just 1 year ago. So again, the rate of genetic change is moving fast. So what is possible? On each chromosome, we measure the value of that chromosome. And so when they multiply, or when they figure out what the genetic value of that animal is, they take the value on chromosome 1 and they add it to chromosome 2 and they add it to chromosome 3 all the way down to chromosome 30. And of course, most individuals have some pluses, but they also inherited some negative traits. But if you could combine the highest value in the Holstein breed for all 30 chromosomes and add them together, notice 14, there's an animal with over $300 net merit just from 14, and 30 also a very high. You would have a breeding value of 6,173. And of course, the possibility of doing that is infinitely small, but it says that there isn't a plateau to genomics or to genetics. Every generation has the ability to go higher. Many when you're a graduate student, you work with Trabolia, fruit flies, and you work with mice lab. You can go through 60 generations of mice in a very short period of time. Now you know why there's so many mice running around your farm is because the generation interval is extremely small. But they measured the milk production in the mouse lab, and every generation, even 60 generations later, they are still increasing. So the rate of gain is there, but there is no plateau in terms of genetics. So here's the opportunity. If we could combine it all, look what we could do to produce a super cow. So again, we talk a little bit about what are some of the more profitable things, and you know these better than I. You want cows that can produce high volumes of fat and protein with relatively low somatic cell. You want animals that are relatively medium in size. You want well-attached capacious udders. You want animals that are mobile and trouble-free in terms of the feet and legs. You want them to calve back easy and breed on time. And you also want cows that can maintain adequate body condition. So the goal of sire selection is to efficiently select a group of sires to use in your herd that will meet your goals that you've established for your herd. And no two operations are the same. One might be marketing livestock or progeny. Some else might be selling to a cheese plant. Somebody else might be selling to a bottling plant. And so there's probably no two herds in here that would have identical selection goals, because depends on exactly what your operation is. And so in some ways, the net merit that Francisco talked about, that's what the average dairy probably has. But there are probably individual items on your dairy that maybe means that you should make a different selection goal than what the national goal is. So clearly, the most efficient way, and I won't spend a lot of time on this because Francisco talked about it, is to use a selection index. But remember, when you select these bulls, you're going to breed this cow today. But that unit of semen is not going to result in product that you're going to sell for several years. And then that animal is going to be in the herd for several years. So the question was, how do we select? When you think about making your selection goals, you need to think about what is going to be the economic conditions on my dairy 4 to 7 years from now. So if you do go in Florida to component pricing, the time to change your selection index is now, because 4 to 7 years from now is when that decision will come to fruition. So if you think that the conditions on your dairy farm are going to substantially change in the next 4 to 7 years, you need to change your selection goals today when you make your purchase of semen. You've seen these indexes. I won't go over those. He talked about them already, but there's significant differences on a couple of them depending on what the index, but they're highly correlated. So clearly the use of a group of genetic young sires will greatly increase the genetic merit in your herd when compared to a group of top proven bulls or even a group of not so elite genomic young sires. Young sire usage, I had a former president of the Holstein Association indicated it. He used to use about 10% young sires. And he did that to use on cows that he didn't know whether she was in heat, or so he used it for problem breeding cows. Today, 90% of all of his cows are bred to genomic young sires. So that's kind of the switch has gone, and you can see just across the country that in 2012, DHIA herds in the Southwest already were averaging over 65% genomic young sires, and I'm sure that rate has increased since then. Here's an important concept that Francisco talked about, and there's a mathematical formula that works it as well. But if you have the reliability of an individual animal is, say, 75%, then the group average of 10 would be 97.5% reliable. In other words, if the accuracy of an individual is 75, the accuracy of that group of 10 sires is 97. So it's the group average equals 1 minus and that whole formula, but that's where Francisco got that formula or whatever. But the actual risk of using a group of 10 genomic young bulls is equivalent to about a 98% reliable bull. So a bull that's got 1,000 progeny is going to be about the same. One progeny as one bull at 1,000 progeny is about the same as using 10 genomic young sires in terms of managing your risk. So that's why we talk very important that you use a group of young bulls. Don't fall in love with just 1 or 2 young bulls. And of course, just because a bull is genomically tested these days doesn't mean that he's superior. You need to make sure that he is, look at the rank that he, for the traits that you're using in your herd. Some of you might have read the February 25th Hort's Dairyman. You talk about hot off the press. Chad Deckow, I think, spoke at this conference a few years ago. He's a professor at Penn State University. And he says a genomic young sire, which because he's lower reliable, he puts this value times the net merit dollars. Daughter proven bull, because their reliability is higher and less risk, he puts this value per net merit dollars. And then a no risk adjustment, so this would be a bull that would be extremely high. The key also is that if the conception rate is at 35%, and that's plenty higher. So if what it says is the higher conception you have, the more you can afford to pay for semen. But he suggests that a Genomic Young sire, because an individual is low reliable, if he was 600 dead merit, you could justify paying $95.46 per unit of semen. Daughter proven bull, because of the higher accuracy, again, their genomics isn't quite as high, but that would put you at $86.55. And the no risk adjustment, I would argue that this is when you take a group of 10 genomic young sires, that accuracy is approaching 98%. You could then justify a 600 merit $112.32. So again, Francisco mentioned about the value that genetics is giving you for the cost. I think most of your semen suppliers would suggest that that price is far in excess of what they're charging for most of the young bulls. So again, based on the values that Chad Deco did in his research, with reasonably high reproductive management, semen from the elite bulls are most reasonably priced compared to bulls of lower genetic merit. So again, a double whammy. You get more the higher you go up the genetic tech. Some other considerations, many registered herds are using a combination of genomic testing, embryo transfer, IVF, and some are also utilizing sex semen. Unfortunately, these young bulls don't produce a lot of semen. Sex semen gives you even less, and so most of the really elite bulls, it's very difficult to find sex semen for those, and so sex semen may or may not work in your plan. So the question is, can a commercial dairyman justify genomic testing? The last 2 months, the Council on Dairy Cattle Breeding have tested 40,000 animals each of those 2 months, driven primarily by commercial dairymen testing the females. So about 3,000 bulls and about 37,000 females each of the last 2 months, and most of those are by commercial dairymen genomic testing. What else does genomic testing do for you that other than genetic selection? What many of you know, we have things called haplotypes that affect fertility. I predict that we're going to keep finding more and more and more of those, and so virtually every animal is going to carry 1, 2, 3, 4 of these haplotypes. Genomic testing, when you genomically test an animal, you automatically get back any and all of those genomic recessives that animal carries. That's part of the, you get the genetic information, but you also get what genomic, and so as Francisco talked about, there are mating programs that avoid inbreeding, but they also avoid mating animals that are carriers of certain things. Can a commercial dairyman justify a sex semen? Again, a little bit depends on your operation. If you're trying to build your herd to a larger herd, you don't want to go out and buy replacements for health reasons or whatever. Again, if you're genomic testing and you want to breed some of the top ones to sex semen to build your cow herd so that you can breed the bottom end of your cows to beef semen or whatever your strategy is, there might be a place in some for those. Commercial herd justify ET or IVF. Again, I think that's maybe a little bit more difficult to do, but again, there may be some places there. But so again, other considerations, the higher conception rate in your herd, the more you can justify for semen. We showed that with Chad's research. Heifers are generally higher conception rate than cows. Given that you've used elite bulls in your herd, your heifers will be the highest genetic values that you have. So it's a win-win-win situation. You can afford to pay more money for semen on high conception cows or heifers. If you're going to make the great progress and create the superstar, breeding the elite animals to elite bulls. I know some herds, I'm originally from California, they would buy a tank of semen, what they called heifer semen, and they would pay a lot more money, maybe 3 or 4 times more for that tank of semen, and they would go to the heifer ranch and would just breed heifers because they knew that it would generate the best to the best, and they were better conception, and therefore they could pay more, and then they would have a tank of semen that they bred their cows that generally they didn't pay quite as much money for. So the question of inbreeding is one of the areas that We had had some hope that maybe genomics would find that truly great unrelated bull. But in general, if you're totally unrelated to the population, those SNPs that that bull has, the system doesn't really know, they're not very well defined, and so finding the great outcross bulls hasn't really materialized. In fact, inbreeding was about 4.5% in 2000, and 2013 it had gone to 6.25%. That's a lot of increase in a fairly short period of time. Not quite as bad as the Jerseys. The Jerseys, some of those are approaching 10%. But the expected future inbreeding in U.S. Holsteins was 4.8%, and then only 6.1% only, but less than that. So it does appear that the future inbreeding is not as increasing as quickly, and it might be leveling off just a little bit. But we certainly do need to pay attention, because we know that there is inbreeding depression as a negative result of breeding closer related individuals. But the use of a large group of sires to sire the next generation, as well as something I'll touch on briefly as a genomic mating program, will help manage this genetic diversity and slow or possibly even reverse some of the effects of inbreeding. So recommendations. First step, need to set the goals for your herd for the traits that are economically important in your herd. It might be different than your neighbors, certainly different than people around the country, but consider the situation on your operation of how it might change 4 to 7 years from now, and now is the time to make the genetic selection change if you think the economics is going to change. I give this talk in Latin and South America, and 10 years ago, they didn't pay anything for protein, and they went and visited with a milk plant, and they said, we probably will have to start changing the way we do it. Those customers changed their selection criteria, and when they started paying for it and testing for it, those herds reap the benefits. So remember, your genetic selection criteria should be not what it is, what you're getting paid today, what the conditions are today, but what it's going to be 4 to 7 years from now. And the more traits you select for, the less progress you will make in any one trait. So loading up your index with lots of different traits, unless it really has a genetic impact, you're probably best to limit the number of traits. And a selection index is the way to go. I would base your selection index, then choose the bulls that you plan to use for the next 3 to 6 months. And then in 3 to 6 months, repeat the process for the following 6 months. Always pick a group of bulls. If you got 1,000 cows or more, probably I would recommend somewhere in the neighborhood of 10 to 15 bulls to breed your cows. And I would recommend that you exceed 75% of your herd bred to young sires. And again, like I say, there are some that are almost 100%. A lot of them have moved up into the 80s and 90s. I believe strongly in mating programs, especially genomic mating programs. Again, when we thought about that pedigree, mating programs traditionally assume that you got an average set of genetics from your parents, and so they said the average inbreeding of this pedigree is 5%. Well, genomically, that animal might be 12% inbred just because of the way the genes that they inherited. So genomic, Chuan Yu, a postdoc at the Council on Dairy Cattle Breeding, came up with a genomic mating program. And a few of the AI companies now offer a genomic mating program. All of them do the traditional mating, but there are starting to available. They get a list of all the animals that have been genomic tested, and they get those animals mated with I think the top bulls that were available a couple of years ago. So they'll be the sire of the individuals, and so you can do a much better job of avoiding inbreeding by genomic mating, because the genomic inbreeding might be very different than what the traditional. So again, regardless of your herd size, extensive use of a genomic young sire will make a significant progress in the economic important traits in your herd that will result in greater profitability in your dairy farm. Any questions? So you might recognize some of the answers to some of those the questions that you had before.